Hello students and welcome back to Microbial Concept. So this is another video on a media that we regularly use in our microbiology labs or while we are performing some practicals, that is MSA or mannitol salt agar. So let's know more about this media. So mannitol salt agar, or it is also written as MSA in short. So it is actually used as a selective and differential medium, which is used for isolation and identification of Staphylococcus aureus, okay, from clinical and non-clinical samples. Now it is for Staphylococcus aureus and not for Streptococcus. Okay, don't get confused. This is for Staphylococcus aureus. And clinical samples, you know, but what are the non-clinical samples which are generally tested for Staphylococcus aureus? So, few examples are like from food industry, where particular food ingredient which is to be added to the food item, it is also tested. Or in case of pharmaceutical industries, the active pharmaceutical ingredient which is further, it has to be used in the particular medicine, it is also tested for pathogens. So Staphylococcus aureus is one of the pathogens which is tested, and there also mannitol salt agar is used. Okay, so the MSA, that is mannitol salt agar, it encourages the growth of certain group of bacteria while inhibiting the growth of others. So it is selective medium which is prepared according to the recommendations of Chapman for the isolation of presumptive pathogenic staphylococci. Okay, presumptive pathogenic staphylococci. Okay, now the recommendation for the pathogenic staphylococci, the composition of the medium is as follows. So it has pancreatic digest of casein, peptic digest of animal tissue, beef extract, sodium chloride, mannitol, the sugar that is fermentable, and phenol red. Okay, so this is how actually the color of the medium it looks. It is reddish due to the indicator that we add, that is phenol red. And after the growth of Staphylococci aureus or Staphylococcus aureus, if it is pathogenic, if it is coagulase positive, then it gives a, or it the indicator, it changes the color to the yellow, and even the colonies they turn yellow. Okay, so this is the basic principle behind this particular media. Now, Staphylococci, they are widespread in nature, and although they are mainly found on the skin, the skin glands, and the mucous membrane of mammals and birds, it is actually sometimes pathogenic, or you can say opportunistic pathogenic. Okay, so the coagulase positive species, that is of Staphylococcus aureus, is well documented as human opportunistic pathogen. So the ability to clot plasma continues to be the most widely used and accepted criteria for the identification of pathogenic staphylococci. Okay, so that's the reason why Staphylococcus aureus is also considered as one of the most potent pathogen which is tested in pharmaceutical industries or dairy or food industry. Okay, the test is mandatory. Now, the staphylococci have the unique ability of growing on high salt concentration media. Okay, the isolation of coagulase positive staphylococci on phenol red mannitol agar is supplemented with 7.5% of NaCl, and it was studied by the Chapman. The mannitol salt agar base, it is recommended for the isolation of coagulase positive staphylococcus from the cosmetic, milk, food, and other specimens, as I told you. Okay, so that's the reason why mannitol salt agar is important, as it is used widely for the isolation and identification of pathogenic staphylococcus aureus. Okay, so this is important. So if you have this for your practicals, then get your concepts clear regarding the principle and interpretation of the results. Now the sodium chloride, which is present in the medium, it serves as inhibitory agent against bacteria on the bacteria other than the staphylococci. And the mannitol is the fermentable carbohydrate, and the fermentation, it leads to the acid production, and thus it is detected by the phenol red indicator. Okay, so when acid is produced, phenol red indicator, it turns the or it changes the color to the yellow. Okay, and that's how we test that, or we interpret that yes, the culture is positive and it is Staphylococcus aureus. Okay, so the Staphylococcus aureus, it ferments mannitol and it produces yellow colored colonies surrounded by the yellow zone, and that's the reason why even media turns yellow. Now the coagulase negative strain of Staphylococcus aureus, they are usually mannitol non-fermenters, and therefore they produce pink to red colonies surrounded by red-purple zone. Now, presumptive coagulase positive yellow colonies of Staphylococcus aureus, they should be further confirmed by performing coagulase test. Okay, so mostly the one which are pathogenic, which which show positive result on the MSA, they should be confirmed, or those particular isolate of Staphylococcus aureus should be confirmed by performing the coagulase test. So you can perform it on a slide or by using a tube. Okay. Now also the organism should be subcultured to a less inhibitory medium not containing any excess salt to avoid the possible interference of salt with the coagulase test. Okay, so this is the important point. Then when you have positive results for Staphylococcus aureus on mannitol salt agar, don't use the culture directly from mannitol salt agar plate. Okay, you have to subculture the culture on a less inhibitory medium which has um, very low salt concentration. Okay, to avoid the interference with the coagulase test. Now, few strains of Staphylococcus aureus may exhibit delayed mannitol fermentation. Okay, it may take time. So, negative results should be therefore be re-incubated for additional 24 hours and then it should be discarded. Now about the principle of mannitol salt agar. So mannitol salt agar, it contains peptone and beef extract which supplies nitrogen, then vitamins, some minerals and amino acids which are essential for the growth of Staphylococcus aureus. It contains 7.5% of sodium chloride which results in partial or complete inhibition of some bacterial cultures other than Staphylococci. Sodium chloride also supplies essential electrolytes for the transport and osmotic balance. Mannitol is the fermentable carbohydrate and the fermentation of it leads to the acid production which is detected by the phenol red indicator, and it helps in differentiation of Staphylococcus species. Now the coagulase positive Staphylococci, that is Staphylococcus aureus, it produces yellow colonies and a surrounding yellow medium, while coagulase negative Staphylococci produces red colonies and no color change of the agar or the medium is seen. Now, if you get a question on this particular media, that is, which are the organisms which are inhibited, then you can give some examples like E. coli, Klebsiella aerogenes, and Streptococci, okay, which has poor to no growth on mannitol salt agar. Okay, Streptococci and Staphylococci. So there is difference. Mannitol salt agar is for Staphylococcus aureus and it inhibits Streptococci. Okay, so this was all about mannitol salt agar. I hope this video is helpful to you all. Do like my videos, do share my videos with your friends, and do subscribe to my channel, and thank you for watching.